The nerve impulse is the sum of the mechanical, chemical, and electrical disturbances created by a stimulus in the neuron. The conduction of nerve impulse may be divided into two phases: resting membrane potential and action membrane potential. Neurons are excitable cells because their membranes are in a polarized state. Neuron cells may be stimulated by physical, mechanical, chemical, and electrical stimuli. The nerve fibers or axon is covered by neural or axonal or plasma membrane. The neural membrane has sodium and potassium ion channels called voltage created or regulated channels. Those channels are opened or closed according to the electrical potential across the membrane. So these ion channels are selectively permeable to different ions. When a neuron is not conducting any impulse, that is resting, the axonal membrane is comparatively more permeable to potassium ions and nearly impermeable to sodium ion. Similarly, the membrane is impermeable to negatively charged proteins present in the exoplasm. Consequently, the exoplasm inside the axon contains high concentration of potassium ion and negatively charged proteins and low concentration of sodium ions. In contrast, the fluid outside the axon contains a low concentration of potassium ions, a high concentration of sodium ions, and thus form a concentration gradient. These ionic gradients across the resting membranes are maintained by the active transport of ions by the sodium-potassium pump, which transport 3 sodium ions outward for 2 potassium ions into the cell. As a result, the outer surface of the axon membrane possesses a positive charge, while its inner surface possesses negative charge, becomes negatively charged, and therefore is polarized. The electrical potential difference across the resting plasma membrane is called as the resting potential. When a stimulus is applied at a site A on the polarized membrane, the membrane at this site A becomes freely permeable to sodium ions. This leads to a rapid influx of sodium ion followed by the reversal of polarity at the site. That is, outer surface of the membrane becomes negatively charged and the inner side becomes positively charged. The polarity of the membrane at the site A is thus reversed and hence depolarized. This electrical potential difference across the plasma membrane at the site A is called the action potential, which is in fact termed as the nerve impulse. At site immediately ahead the axon, that is site B, membrane has a positive charge on the outer surface and a negative charge on its inner surface. As a result, a current flow on the inner surface from site A to B. On the outer surface, current flows from site B to A to complete the circuit of current flow. Hence, the polarity at the site is reversed and an action potential is generated at site B. Thus, the impulse, that is action potential, generated at site A arrives at site B. The sequence is repeated along the length of the axon and consequently the impulse is conducted. The rise in the stimulus induced permeability to sodium ion is extremely short-lived. It is quickly followed by a rise in permeability to potassium ions. Within a fraction of a second, potassium ion diffuses outside the membrane and restores the resting potential of the membrane at the site of excitation, and the fiber becomes once more response to further stimulation. So this is the diagram showing this action potential and number impulse. A nerve impulse is transmitted from one neuron to another through a junction called synapse. And a synapse is formed by the membranes of a presynaptic neuron and a postsynaptic neurons, which may or may not be separated by a gap called synaptic cleft. This is the in the diagrams you can see the presynaptic neuron, postsynaptic neuron, and synapse. And there are two types of synapses, mainly two types, that is chemical synapses and electrical synapses. In this diagrams you are looking the presynaptic neurons, synaptic and postsynaptic, and this chemical synapse and electrical synapse. Chemical synapse is more common in human beings. At a chemical synapse, the membrane of the pre and postsynaptic neurons are separated by a fluid-filled space called synaptic cleft. Chemicals called neurotransmitter, that is acetylcholine, are involved in the transmission of impulses at these synapses. The axon terminals contain vesicles filled with these neurotransmitters. When an impulse or action potential arrives at the axon terminal, it stimulates the movement of the synaptic vesicles towards the membrane, where they fuse with the plasma membrane and release their neurotransmitter in synaptic cleft. The released neurotransmitter bind to their specific receptors present on the postsynaptic membrane. This binding opens the ions channels, allowing the entry of ions which can generate a new potential in the postsynaptic neuron. The new potential developed by may be either excitatory or inhibitory. So this is the whole process of chemical synapse, in which this neurotransmitter is now the electrical synapse. Electrical synapse are rare in our systems. At electrical synapse, the membrane of pre and postsynaptic neurons are in very close proximity. The electrical current can flow directly from one neuron into the other across this synapse. This is the electrical synapse. It is found first in crayfish, then coelenterate, then annelid, then mollusk, arthropod, and fish. So it is rare in human beings. And at electrical synapse, the membrane of pre and postsynaptic neurons are in very close proximity. The electrical current can flow directly from one neuron into the other across this synapse. Transmission of an impulse across electrical synapse is very similar to impulse conduction along a single axon. Impulse transmission across an electrical synapse is always faster than the across a chemical synapse. This is the CNS in diagrams. Okay, so thank you very much.